So I wanted to start with this nice quote from a paper from Susan Solomon from New York, that iPS cell authentication and quality underlies their value at the frontier of biomedical advances. And they state that researchers need the assurances of strict quality control, conscientious tracking, and definitive identification to produce studies that yield reproducible data and valid results. Now, the National Institute of Health in the USA has recognized this issue, especially surrounding iPSC authenticity, as a concern about reproducibility in cell culture studies. And it's also worth mentioning that a number of large journals, such as Nature and Cell Stem Cell, have also had a renewed focus on quality, about putting new standardized practices into place in order in the future to go and publish in their journals. I want to state that cell line derivers and suppliers need to adopt the highest quality standards and verify and report the properties of these PSCs. A key step towards this goal will be stem cell researchers, such as yourselves, reaching a consensus on appropriate standards and best practices, especially in collaboration with these international consortia. So at STEMCELL Technologies, we've defined 5 pillars of cell quality for PSC research. The first being morphology, so how these cells visibly look down the microscope, the epigenetic landscape or the methylation status of these cells, the validation of pluripotency, the monitoring of genomic integrity, and also analyzing gene and marker expression. Now, the interesting thing about cell quality is that if you were to ask a number of different pluripotent stem cell researchers what a quality cell or cell line is, I think we'll probably get a different answer every time. And even if you go to members, even within the same laboratory, everybody has a different answer. We also have to be very realistic about the limitations that are put in place on quality standards, such as the time that we want to spend analyzing the quality of these cells, the amount of money that we want to put into it, and think about what return we're actually getting back. But what we do believe is that cell quality attributes should be considered by everybody that's working with PSCs, and not just those researchers that are taking these cells to the clinic. So we'll begin with morphology, and we believe that this is actually a very sensitive indicator of cell quality. It's good because it can be checked frequently, daily for example, just by checking the cells under the light microscope. However, this is quite a subjective method, and it's largely based on experience, and once again, every different lab member, when looking down the microscope, will probably have a different opinion of what a good versus a bad morphology is. Morphology can also differ depending on the culture conditions that are being used, so many different types of pluripotent stem cell media that are available out there, matrices, passaging reagents, and also the stem cell state that's being cultured, such as primed pluripotency or the naive-like pluripotent state. Now at STEMCELL, we passage these cells as small aggregates. We like to keep them as clumps, which means that these grow as colonies, as 2D colonies in the dish. And as a result of that, we see this morphology, which we'd like to see, this very round, tightly packed cells with a very large nucleus to cytoplasm ratio. We like to see smooth edge colonies that are very circular. And we also like to see multi-layering in the center of the colonies. There's this phenomenon with teaser media where the cells in the center of these colonies divide a little bit more rapidly, which means they're a little denser in the center, and you start to see stacking. So you start to see this phase brightness under the phase microscope when these cells are ready for passaging. Now, in order to test if morphology is in fact a very sensitive indicator of cell quality, we set up an experiment. We made 2 different media formulations. One is mTeSR1, which you may have heard of or you may have used. And this is for the maintenance and expansion of undifferentiated pluripotent stem cells. We also made a version of mTeSR1 that was suboptimal, so it had largely reduced levels of FGF in the media. FGF, a key cytokine in the media to maintain this undifferentiated, proliferative pluripotent state. Now, we work with a morphology scoring system where we like to analyze the morphology of our cultures based on a scale that goes from 0 to 4. 0 being a very, very poor quality culture, and 4 being very high quality culture. And what you can see is when we withdraw FGF from the medium, this suboptimal medium, decreases in morphology score after 3 to 4 passages. So it actually took some time to see this deterioration of morphology under the microscope. Now, even when we actually took these poor quality or poor morphology cells and ran the markers, or the undifferentiated markers, TRA160 and OCT4, we would have expected these markers to potentially be lower. Okay? However, that was not the case. And we actually saw very similar marker expression between mTeSR1 cells and suboptimal cells. So whether these markers are actually a very sensitive indicator of cell quality is a question that should be asked. And it seems that in this case, morphology appears to be a more sensitive indicator than this marker expression. Now, as I mentioned, morphology can differ between different culture systems. Now, there's many different culture systems available now, and as we go through the evolution of these systems, starting in this system here, this is the H9 cell line, and the photos that I'm about to show you on subsequent slides are all different images of H9s in different culture systems. So here you can see H9s cultured in knockout serum replacement media on inactivated mouse embryonic fibroblasts, or MEFs, or feeders. And these feeders can be seen in the background of this culture plate. These are the long spindle-like cells that are providing some structure to these colonies as well as nutrient support. Okay. Now, these colonies in a feeder-based media are actually fairly small, however they're very, very densely packed together. Okay, so that's a key morphology that we noticed in the system. Now, with the evolving system over to the mTeSR1 system on Matrigel, this is what a colony looks like in a clump passage system. So here you can see these colonies are actually much larger. We're starting to see phase brightness in the middle of the colony, as well as a removal in the background of all of these spindle-like feeder cells. Okay, so this was the first established feeder-free system. Now, many people we've noticed are actually starting to passage these cells a little bit differently. Okay? And one very common method of passaging now is single cell passaging. So that's every time you want to passage these cells and put them into a new daughter flask, we are singularizing all of the cells that are in that population. And the morphology looks different. So here you can see again H9s on mTeSR and Matrigel, but this time with different passaging method. Okay, this was with acutase. And here you can see we've lost this colony-like morphology, and we're starting to see a monolayer-like morphology spreading across the bottom of the plate. And finally, this is an image of H9 cells that are cultured in a media that we provide from STEMCELL called Rosette. Rosette is a media for the reversion of the primed-like state to a naive-like state. Okay? Now, in this instance, you can see these colonies look a little bit different. They're actually domed, so they almost look a little 3D coming off the surface of the plate, and they're very refractive around the edges. And these look very similar to what mouse embryonic stem cell lines look like. This is how they're cultured. Now, we're not saying that any of these morphologies is bad. I'm not saying that any of them are good, but they are certainly all very different. Okay? So which one is good, which one is bad, if they are, is an interesting question to be asked and something to think about moving forwards. So the next quality trait I wanted to touch upon is gene and marker expression. Now, many PSC researchers check these cell surface antigen expression on the surface of the cells. These are markers such as the SSEAs 3 and 4, or the TRA markers, TRA160, 181, TRA254, alkaline phosphatase, that kind of thing. Now, this staining can be done very regularly, but again, going back to this previous slide, is it actually indicative or reflective of quality? We can also look at relative gene expression. So we know that there's developmentally regulated genes that are expressed similarly in this cell type. So for example, NANOG, OCT4, SOX2, etc. Another method that researchers are now employing quite frequently is Rna-Seq. And this is a nice method, is that enables us to look at the RNA profile of these cells, or the gene expression profile of these cells, in different conditions. So for example, we can take the same cell line and look at the gene expression profile between different culture systems. Or we can take different cell lines and see how they behave in the same culture system to see what the variability is. This is a nice technique. However, Rna-Seq is still quite expensive and it takes some time to get the data back. Now going back to this cell surface antigen expression, this is a nice diagram explaining how the cell moves from a pluripotent state into a differentiated state. Okay, so here we have a yellow, undifferentiated pluripotent cell here, sitting at the bottom of this hill. Okay. And in order to differentiate into a terminally differentiated cell type, this cell has to climb this hill, changing multiple different, what seems like different energy states, and fall off the edge of this cliff at the end where it then differentiates. Okay. So as this cell climbs this hill, you can see that the first marker that it loses is SSEA3. Okay. So it seems that SSEA3 is potentially the most sensitive marker to measure the undifferentiated stem cell state. Once it loses SSEA3, it then loses TRA160. Okay, so TRA160 may be a little less specific to the undifferentiated state. And then once we have lost both of these markers, the cell then commits and differentiates. Okay, so this is a model of how this system works and what we're actually measuring when we're using these markers.
Yes. Yeah. So essentially this is a marker of the prime iPSs.
Yes. Yeah.
Not the naive.
I'm not sure whether SSEA3 is on naive. I actually can't remember. Yeah.
This figure describes a prime state iPSs or ESs.
For the majority of this presentation, it's focusing on the prime state. Yes, yeah. What is interesting is, and I can't recall if this is SSEA3 and SSEA4, but it's estimated that one of those markers is actually negative in 1% of all humans. Okay, so that means, theoretically, that 1% of iPS cell lines that are being generated are actually SSEA negative. Okay, so we can't even use this marker in all cases, so something to think about, yeah. So next, moving on to pluripotency. Now, what do we mean by pluripotency? So the very definition of this term and this state is a cell that has the capacity to differentiate into all three of the embryonic germ layers to try to validate pluripotency versus our kit. Now, in this particular experiment, we went to test H9 human embryonic stem cells. So H9s are the most commonly used human embryonic stem cell line in the world. In this heat map of the gene expression array that we have here on the left, red is high expression and green is low expression. And you can see that for the undifferentiated cells, we're getting nice positivity for the undifferentiated markers. Now, when we took the EBs, these were generated using a 10-day protocol in serum-containing media, driving differentiation. You can see that they differentiated to the ectoderm lineage very well, whereas they did not differentiate to mesoderm and endoderm. Now, when we run these cells using the STEMdiff Trilineage differentiation kit, you can see we get very nice patterning of ectoderm markers, mesoderm markers, and endoderm markers with the three different media. So where we did not manage to validate pluripotency with the EB assay, we did with the STEMdiff Trilineage differentiation kit. Okay, so next we want to move on to the genomic integrity of these cells. So unfortunately, one thing that we do know about human embryonic stem cells is that if you culture them for a long enough time, and experiments have been done to take them out to 100 passages, 200 passages, that kind of thing, is that we do find that they acquire karyotypic abnormalities. Now, we also see that a difference in behavior of these late passive cells to these early passive cells, and we see decreased population doubling time, so faster growing cells, and increased plating efficiency, so they're sitting down on the surface of the plates better. So these characteristics have been linked to these karyotypic changes observed by G-banding, and these changes occur in a non-random and sporadic manner. So we're seeing a growth advantage with these karyotypically abnormal cells, and these particular abnormalities that we recurrently find in PSCs are also observed in a number of human cancers, such as teratocarcinoma. Now, this study that you can see here is from the International Stem Cell Initiative, dating back to 2011. And in this study, they analysed 120 human embryonic stem cell lines. And for all of these lines, for the different labs that were maintaining them, they analysed the karyotype at early passage and also late passage. to see if there were any differences. Now, out of these 120, 66% were normal in both instances. 20% went on to gain an abnormality. 12% were abnormal at both early and late passage number, and 2% actually managed to lose an abnormality. So there's a definite trend for abnormality gain. And out of all of these abnormalities that were detected, the acquisition of chromosomes 1, 12, 17 and 20 happened again and again and again and again and again. As well as these large structural changes, they also used SNP array to try to identify any small structural variants. That's anything under 5 megabases, which is the limit or the threshold of a G-band. Now, in 22 of the normal lines that came back normal, they were actually harboring a duplication of this region in 20q1121. Okay, so this is a very small abnormality, which was actually subsequently found to confer a very strong selective advantage to these cells. Now, this is quite worrying because you cannot see this abnormality on a G-band. Okay, and G-banding is, of course, the standard way of analysing the genomic integrity of these cells. So how many projects out there have been working with cell lines that actually harbour this 20q abnormality without knowing so? So here you can see a paper that compiled a list of all of the abnormalities from many different labs, and we constructed an ideogram. And what you can see is that green is the acquisition or the gain of genetic material, red is the loss of genetic material, and in blue we have balanced structural rearrangements. Now, the immediate thing that you can see from this ideogram is that there's a lot more green than any other colour. Okay, so what is happening is that pluripotent stem cells appear to be gaining genetic material in order to help them live better, grow better, sit down better. Okay. So what does this mean and why is it important? So if we go back to the very basic question of what is a stem cell can do 3 things. It can self-renew, produce an identical copy of itself, a daughter cell. It can differentiate, or it can die. Now, the whole pluripotent stem cell workflow is actually a very highly selective process. So if you think about where we go from and when we go to, so we start at the beginning with reprogramming somatic cells. Okay? So we reprogram these cells and we get some clones back, and we need to pick those clones. Okay? But we only pick the good ones. We only pick the ones that look nice, that have good morphology. So, you know, what the difference is between those good-looking ones with our own eye versus the bad ones, we don't really know. Secondly, we pick these clones, then we place them into long-term culture. Okay? And every single time we want to passage these cells, we're using a passaging reagent, we're plating these cells into the next flask or the next plate. Some cells survive, some cells die, some cells don't sit down. So we are selecting every single time for a certain subpopulation of cells. And what they are, we don't know. Okay. And then finally, at the end of this process, we want to differentiate these cells. That's why we're using these cells, of course. Right? Now, these differentiation protocols are not 100% efficient. So some of them differentiate and some of them don't. And of course, we are selecting for the ones that are actually differentiating. So by the time that you've got to the end of this whole process, we've done a lot of selection of subpopulations without even realizing it. And what we've selected for, we don't know. Okay? So these abnormalities have been linked to reduced cell cycle times and an increase in the rate of cell attachment, like I mentioned. This leads to a decrease in the rate of differentiation and a decrease in the amount of cell death. This increases the rate of self-renewal. So these cells are now growing and surviving better when they're abnormal. So we wanted to test this hypothesis that abnormal cells differentiate differently. Okay, and this was an experiment that we set up in-house at STEMCELL, where we took the 1C iPS cell line, and this is an iPS cell line that we generated in-house, and we waited for it to get an abnormality. And sure enough, eventually we found an extra copy of chromosome 12 in this cell line. Now we managed to clone out a cell with trisomy 12 and clone out a normal cell, and then we generated 2 new cell lines. So they're both 1Cs, they both have the same genetic background. However, one of these lines is now 100% normal, and one of these lines is now 100% trisomy 12. So we wanted to see if there was any difference in the patterning of differentiation. So we drove these 2 lines through endoderm differentiation protocol using our STEMdiff definitive endoderm kit, and left it in this kit for 6 days. Now, by the time we get here, we would expect all cells to be OCT4 negative, and we would expect most of the cells to be SOX positive. Now, if they weren't, what could happen is if we place these differentiated cells back into mTeSR and matrigel, we're thinking maybe something might grow if there's still undifferentiated cells in there. So that's what we wanted to see. Now these are the facts plots. So this is what the whole process looks like. We have the control cells on the left, the wild type, and we have the abnormal cells on the right. And here you can see the undifferentiated cells. So this is before we started the experiment. We have OCT4 on the x-axis and SOX17 on the y-axis. So you can see nice high positivity of OCT4 in both cases. This is exactly what we would expect. Now, as we go through this differentiation protocol, this is what we would expect to see. We would expect to see cells moving from this quadrant here, OCT4 high, SOX17 low, to this quadrant up in the top left, SOX17 high, OCT4 low. Okay? So let's go through the 6 days and see how the 2 different cell lines move. So day 1, day 2, 3, 4, 5, and 6. Okay? So we looked at these results and we said, actually it worked really well for both lines. Okay, so there's not really a deficiency in any differentiation. You can see 94% in the top left here and 95% in the top left here. So not really any difference in patterning in differentiation to endoderm. However, there was something interesting on this graph which we found after looking at it a little bit harder. Can anyone see what might be interesting on this graph?
The small fraction of the SOX3, SOX17 negative, SOX17 low, or positives.
Exactly. These cells here. Okay. So 0.6%, which is very, very low, granted, were still OCT4 positive in the abnormal line, whereas only 0.06% were positive here. So that's an increase in 10 times. Okay? Now it still sounds low, but if you can imagine you're implanting 1 billion cells into a patient, okay, and 0.6% of them are OCT4 positive, we've got a problem. Okay, so this is an undifferentiated phenotype that's being put into a human. Okay, so that's worrying. So we took all of these cells, and we took all of these cells, and we placed them back into mTeSR and matrigel to see if anything grew. And this was the result of the control line. So here you can see the endoderm cells going into mTeSR at day 1. Day 2, morphology changes a little. Day 3, we're seeing a lot of cell death and a lot of blebbing, cells floating off. And by the time we got to day 4, nothing survived. So mTeSR medium could not bring back, could not maintain any undifferentiated pluripotent cells in this culture from the control line. However, when we place the abnormal cells, the abnormal differentiated cells, back into mTeSR, we got something very interesting. And by day 6, of course, you can see we have a very nice, undifferentiated stem cell colony that is being expanded, and we also stained, we could see that it was OCT4 positive. So this is very concerning for anybody going to the clinic if they're not routinely monitoring the genomic integrity of their cell lines. Who uses one of these? So we have acutase on the left. I used to use them. Acutase on the left, triple select on the right, trypsin. Yeah? Quite a few. Yeah. So I guess if there's one message, or take-home message today, I'd like to say is please, please stop using these. Okay? Please. For the routine maintenance of pluripotent stem cells, this is not good. So we know, we're putting a lot of work into analysing the difference between clump passaging and single cell passaging of cells. Single cell passaging using acutase, triple, trypsin, that kind of thing, is the #1 factor that we are doing every day that can increase the risk of acquiring genetic abnormalities. Okay? So here you can see here from a paper on the left, again from Jean Loring, an experiment where they took human pluripotent stem cells that were initially cultured in a feeder system and put them into 4 different conditions. 2 of the conditions were on feeders and 2 were in a defined feeder-free system. Now within each of these, we had mechanical passaging and we had enzymatic passaging. Mechanical passaging is clumps, okay? It's taking aggregates. Enzymatic passaging is single cell passaging. Okay? And if we can see the results here, you'll struggle to see the x-axis, but this passage number, I think it actually goes up to 160, so very high passage numbers. You can see immediately a dramatic increase here with the blue and the green lines, okay, very early passage numbers. And this is the number of duplications that are appearing in these cells. Blue and green correspond to both the feeder and feeder-free system, enzymatic passaging both times. Okay, and this is something that we are mirroring with all of our in-house testing, looking at abnormalities ourselves. So why is it that single cell passaging leads to an increase in karyotypic abnormalities? You know, it doesn't really make sense. Say that again, sorry.
What is the difference for figures What's the difference
in the 4?
Yeah.
Okay. So top left is the number of duplications. Top right is the number of deletions. Yeah. Bottom left is the length of the duplications, and the bottom right is the length of the deletions.
So how quickly is several passages?
So this differs, and I'm about to explain actually the mechanism that we're looking at here, but it does seem that you can go from a very low percentage of abnormal cells to a very high percentage of abnormal cells in as little as 5 passages. Okay. So it is quite a rapid uptick and takeover of these cells. So let's say we're starting with one well pluripotent stem cells, and here you can see these colonies. I apologise, this isn't in the handout. I made this on PowerPoint in 5 minutes yesterday, but I think it gets the message across. So here we have lots of normal, wild type, nice human pluripotent stem cell colonies. Okay. Now, in long-term culture, at some point there's going to be an abnormality that's going to occur. Now, in clump passaging, this abnormality we find actually stays localized initially to one clump. Okay? So here you can see one clump has gained this abnormality. Now, if we do clump passaging and we're keeping all of these colonies as small aggregates, what we're doing is we're breaking up each of these colonies into maybe 4 small chunks, 8 small chunks, something like that. Now, if we're working with very high split ratios, which we do with clump passaging, such as 1 in 50, the chance of getting one of these 4 chunks that this red colony is being broken into going into a daughter well of maybe 6 wells is actually very, very, very low. Okay? And most of the time, we're going to be getting this version of passaging. And every now and again, we are going to pass one of these clumps into a well. And we're going to get the abnormality being passed over into the daughter well. However, the chance of getting all 4 of these in one well is extremely low. The chance of getting one of them, still low, but possible. Okay? So it's going to stay localized again. And this is why, if we do clump passaging and we have an abnormality, that abnormality rate stays quite low. Now, eventually, it's like it's a game of chance. Okay? Eventually, we're going to get one passage where, by chance, this aggregate is going to be broken up into 4 or 8 pieces, and maybe all of them might go into the next well. Okay, this is where we get takeover, and this takeover happens very, very rapidly. Okay, but on average, it takes a long time. Now, let's compare this to single cell passaging. Okay, this is what happens. We are getting individual single cells interspersed everywhere into the next passage. Say it's like sneezing. Okay, it goes everywhere. It's being interspersed. And what happens is, of course, these abnormal cells we've already looked at, they grow faster, they clone better, and they differentiate less. Okay? That means that they are out-competing their normal neighbours. We then get rapid takeover of the single cells that are abnormal. Okay? This is the mechanism behind why clump passaging keeps these abnormalities levels low. Okay? Single cell passaging does not increase the rate of the mechanism that is inducing these abnormalities. We don't know what the mechanism is yet. I'm sure we will, but we don't know it yet. And until that point, we're not going to be able to stop it. What we can do is alter how we're passaging these cells to limit the amount of abnormalities that we're gaining. Okay. And I think that something that's worth mentioning is, and I remember, working with these cells back when I was doing my research, and it's something that we don't really like to talk about, okay? Because we know we should be karyotyping these cells, we know we should be sending them up for G-band. But I think that one thing that we forget is that these abnormalities are not happening because we are bad researchers. Okay? It's not that we're looking after the cells wrong. We're still doing the work, and then maybe an abnormality comes in, and we're ashamed of ourselves because we have no abnormality. We're going to have to throw the cells away, and all of the work is wasted. And someone else in your lab doesn't have an abnormality. It doesn't mean that they're looking after the cells any better than you are. It just means that by chance your cultures have gained an abnormality. And I think we need to be talking about abnormalities more, because at the moment they're being hidden in research and they're affecting the results that are being published. Okay? So I think this is an important issue, and I think it's something that we could all do to potentially improve the situation. So there's many different methods that we can use to analyze the genomic integrity of your cells. As I mentioned, the most common is G-banded karyotyping. This is good because it can detect quite a lot of different types of abnormalities, such as inversions, duplications, deletions, translocations, aneuploidies. But as we mentioned before, the limit is 5 megabases, so it can't detect submicroscopic abnormalities. If you want to detect them, you have to couple G-banding with a microarray, so ACGH or SNP array. Okay. Now these are quite expensive, so by the time we've done 2 of these together, we've probably spent about $1,500, okay, just characterizing one cell line. There's also FISH analysis that can be done. Apart from FISH, only looks at one very, 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 very small region, so you can't get a lot of data. And of course now, there's whole genome sequencing methods. However, this is still very expensive, even though it is coming down in price, and still takes about 3 to 4 weeks to actually get a result back. So the message of this slide is there's no perfect method. You have to use a couple together if you really want to get a good idea of what's going on. So it's an interesting question. How frequently should we monitor genomic integrity? Okay. So, hypothetical situation, we start with a cell line that we characterize for the lab, and we bank. and under characterization, this cell line came back as 46XY normal. Okay. We then start doing our experiments, and these cells go into culture for a long time. And we're getting data all the time, we're doing all of our different experiments because we want to publish. Okay? And maybe we're doing a PhD, okay? And 4 years later, we have all of our data, and we want to publish our data in a journal, so we write the paper, okay? And we go to the journal and we say, here's all the data I want to publish, and they say, you need to give us a G-band, okay? You've got to give us one G-band. Right, okay, so let's go do a G-band, and it comes back, and this G-band is trisomy 12. Okay, what does this mean? Okay, if you've not done this for 4 years, did this abnormality happen here as you were publishing? Did it happen here right at the beginning, or did it happen somewhere in the middle? We don't know. Okay, and we've already looked and found out that this abnormality is very powerful. Okay, so if you're doing a CRISPR-Cas9 gene edit, for example, and we're trying to model a disease, and this disease is like a one single base pair change that we've managed to change with CRISPR-Cas9. Okay? Are we modeling that genome edit? Or are we modeling the whole acquisition, so that's ectoderm, mesoderm, and endoderm? And the most historic, or what we term the gold standard way for measuring pluripotency, is to use the Teratoma Assay. Okay, so the Teratoma Assay is one where we take a cell population which we consider to be pluripotent and inject it into immune-compromised mice. The histology of the resulting tumours that develop are then analysed for the presence of all 3 germ layers. And you can see a very nice histology slide on the right here, where we're seeing evidence of all 3 of these germ layers. So we have muscle at the top here, cartilage here, so these are both indicative of mesoderm. We have this neural tube forming here for ectoderm, and we have these endodermal tubes forming here. So this is a nice slide that demonstrates all 3 lineages. There are, however, some drawbacks to this assay, and the big one is that it's very inconsistent from lab to lab to lab, so no 2 labs actually do this assay the same, it seems. A lot of inconsistency between where we're injecting the matrix that we're using, the number of cells that we're injecting, how long are we leaving the cells in the mouse? Is it 2 weeks, 6 weeks, 12 weeks? It's going to give you a very different result every time. The age and strain of the mouse, the passage number of the cells that you're testing, etc. So there's a lot of variables that are involved, and this hasn't really been standardized. Unfortunately as well, not all histology slides look like this one. So this is an image of a slide taken from a paper from Damian and Andrews back in 2016. And here you can see a tumour containing cystic and tube-like structures made of undifferentiated cells with condensed nuclei and very scant cytoplasm. It's very, very difficult to characterise what these cells are. And we're not going to be able to without putting a lot more work into characterising what, extra characterisation steps that need to take place. So because of these issues, people are now turning to different methods of assessing pluripotency. And one question that we like to ask researchers is, how do you assess the pluripotency of the cells that you're working with? And I would say that probably the most common answer that we get back is, well, I measure the pluripotency markers of my cells. That's a very common answer. Now, it is true that OCT4, NAMOG, and SOX2 do correlate with the teratoma forming potential of PSCs. However, there is no marker that can predict or is indicative of whether a cell can differentiate into all 3 germ layers. Okay? There is no such thing as a pluripotency marker. They do not exist. These are markers of undifferentiated cells. They are undifferentiated cell markers, not pluripotent markers. Okay? So we can't do this to validate pluripotency. Other researchers are turning to computer prediction models such as Pluritest, that originally came from Jean Loring at Scripps, and also Scorecard. And these are very nice bioinformatics tools. However, they do largely rely on feeding undifferentiated cells into the system and trying to be predictive of what they're going to do, and it doesn't always work. Ideally, what people would do is differentiate these cells and then run them on scorecard, but it seems that that's not actually what people are doing. And finally, we have in vitro differentiation methods, be it spontaneous or directed. So the most common assay, and one that's been done for a long time, is embryonic body formation, where we scrape aggregates, put them into suspension in a serum-containing media to try to drive differentiation. We can direct differentiation to things like neurospheres, cardiac bodies, and there's also monolayer differentiation methods that are being created as well. And I just wanted to focus on these methods over the next couple of slides with a kit that we've created at STEMCELL called the STEMdiff Trilineage differentiation kit. So this is a kit that is has been constructed in order to validate the pluripotency of your cell lines in a very simple manner. So we have 3 media bottles, as you can see, ectoderm, mesoderm, and endoderm. And in order to validate pluripotency, we take the cells that we want to analyze, seed them down as single cells at day 0 into N-1 medium, and then start feeding daily at day 1 with the corresponding media. Mesoderm and endoderm is completed at day 5. Ectoderm takes 2 days longer. This is in order for PAC6 to switch on high enough for us to detect it, and OCT4 to turn off. Now when you get to day 7, you can assess these cells for the lineage-specific markers. You can use any assay of your choice, be it flow cytometry, ICC, you can even do transcriptome analysis if you wish to. And here's some data from this kit. So here you can see we tested 5 independent pluripotent stem cell lines with an N of 13. And you can see that we chose 1 marker per lineage, so PAC6 for ectoderm, T or brachyury for mesoderm, and SOC17 for endoderm. And you can see that in all cases, or pretty much all cases, we're getting very high efficiency of differentiation to these different lineages in over 80%. When we add a different marker in for each lineage, so nestin for ectoderm, NCAM for mesoderm, and FOXA2 for endoderm, we're getting nice dual positivity across the board for ICC. Now, after discussing with a number of different groups that collectively looked at this data, we estimate that the coverage of these chromosomal regions here cover about 75% of all reported abnormalities. So here's some results from the kit. We've actually developed an online tool, a bioinformatics tool, where once you've run your qPCR, you can just copy and paste your CT scores straight into our tool, okay? And it's going to make all the graphs for you, and it's going to say if anything's abnormal. Okay? And this is what the graphs look like. So here you can see the acquisition of the long arm of chromosome 1, and it's stuck itself onto chromosome 21 here, and you can see the increase from 2 copies up to 3 using our kit. Here's a deletion of a part of chromosome 10. Again, decrease from 2 copies down to 1. This is a very easy one to spot on the GBank, trisomy 12, the one we discussed before, an increase from 2 copies up to 3. And then finally, this is the interesting abnormality that we discussed before. So this very, very small 20q1121, this is the one that's very, very, very strong. It's conferring a very strong selective advantage, but you cannot see it on the GBank. Okay? When we sent these cells off for G-banding, we got a normal karyotype back, 46XY, 20 out of 20 cells that were analysed. However, when we ran this sample on the qPCR kit, you can see we went from 2 samples in the control up to 3 samples. 2 copies in the control up to 3 copies. So we went back to the people that karyotyped this and we said, hey, we think this is abnormal. Can we try to find it? So we did some fish analysis. And with the fish, you can see that we have 2 probes, a green one, which sticks to the short arm of 20, and a red one, which actually sticks to a gene called BCL2L1. BCL2L1. This gene resides in this region. Okay, and this gene is the only gene so far to date that we found that we know actually increases the rate of stem cell growth, alters differentiation patterns, increases cloning efficiency. Okay, so this gene has been linked to that. And sure enough, once we sent this off for FISH analysis, you can see 2 green spots of 20p and 1, 2, 3 copies of chromosome of 20q1121. Okay? So using our kit, we managed to validate this acquisition of this very small region for a very low cost, whereas with sending off for alternative methods, we had to do a G-bound and a FISH to actually find it. So some benefits of this kit: it's very targeted, as we mentioned. It's been designed to detect the majority of the abnormalities that are being found in PSCs. It's quick, we go from cells to results within a day. It's very cost effective, so it's low cost per sample. You can actually run 20 samples on one kit. You get a lot of data. This enables the frequent screening. So we're actually doing this assay in-house now, all the time. So we say, every 10 passages maybe, every 20 passages. We're actually doing it all the time. So it's been very interesting to see the patterns that are actually occurring with the abnormalities. And it's also convenient. So as I mentioned, we have an online tool for streamlining data analysis and interpretation.